Good morning. So I'm going to talk a bit about the malaria vaccine trials that we're running from Oxford. So this is a pre-erythrocytic malaria vaccine programme. And for those of you not familiar with the Jenner Institute, we are a viral vector vaccine group focusing on technology development and the application of that to a variety of different diseases. So we do everything from vector design through to antigen selection, through to preclinical studies and clinical trials both in the UK and in populations that we hope to target with our vaccines. We think we're one of the busiest non-profit vaccine institutes globally. So we heard a good introduction yesterday from Bernadette on the issues surrounding the development of a malaria vaccine. And as was mentioned yesterday, the progress of the malaria vaccine field is led by two main candidates. So firstly, RTSS, which is the particle vaccine from GSK with their AS01 adjuvant. That's just completed phase 3 trials in 15,000 infants, giving around 50% efficacy in 5 to 17-month-old children. But disappointingly, in the age that we would like to target with a malaria vaccine, so 6 to 12-week-old infants, that efficacy dropped to 30%. And again, the duration of efficacy in this age group is rather short, perhaps not lasting more than a single malaria season. But we do know that for RTSS, efficacy is mediated by high-titer antibody responses to the surface protein of the malaria sporozoite, which is circumsporozoite protein, or CSP. The other leading approach in the field are whole sporozoite vaccines, so the so-called PFS-PZ vaccine. And you can achieve 100% efficacy against malaria infection with this approach, but you have to give between 4 and 6 intravenous doses of over 100,000 sporozoites. So for those of you who don't work in malaria, that equates to around 4 mosquitoes per dose. And you have to dissect those mosquitoes, take the head off, pull out the salivary glands, pull out the sporozoites, wash them, purify them. All of this has to be done aseptically because you're going to administer these intravenously, eventually to very small children, somewhere in the middle of Africa, and of course they need to be cryopreserved as well. So we know that efficacy with this approach is mediated both by antibodies and T-cell responses. It's very safe, it's very well tolerated in vaccine recipients and meets the regulatory standards set down by the FDA. The big question really is whether this approach is either logistically or financially feasible. And although efforts are being made by Scenario to make separation of sporozoites from mosquitoes into a high throughput process, whether that will ever be really achievable on the kind of scale that you need to do to vaccinate large parts of Africa, I think is up for debate. So, as Bernadette discussed yesterday, there are several possible targets within the malaria life cycle for a vaccine. The most difficult target is the blood stage, where you have many millions of parasites within the blood and you need extremely high titers of antibodies to have any effect on clinical disease, as demonstrated in mouse models. You can also target the sexual stage of the life cycle, and this is now receiving more prominence because although it confers no particular benefit to the individual in terms of generating herd immunity and reducing rates of transmission across Africa, an element of a multi-stage vaccine that had a transmission blocking element would be a significant advantage. And then RTSS targets this stage, the sporozoite stage of the malaria life cycle. This is where we would use antibodies to try and block the introduction of the sporozoite into the liver. And the problem with this stage is that although it's very small numbers of parasites that you need to eliminate, it's a very small window, somewhere between 30 minutes and an hour, that it takes the malaria parasite to travel from the skin to the liver. So this is why we focus on the pre-erythrocytic stage of the malaria life cycle, and it has a number of attractive features. Firstly, this stage of the malaria life cycle is between 5 and 7 days. And although the liver is a large organ and you're looking for a relatively small number of parasites, the fact that they're within hepatocytes that express MHC class 1 makes this an attractive target for CD8 T cells induced by vaccination. So our vaccine is based on the TRAP antigen from the pre-erythrocytic stage of the malaria life cycle. Apologies. Okay, so TRAP is the second most abundant protein at the sporozoite stage, and it's essential for invasion. So we take the TRAP protein and string it to and connect it to a multi-epitope string of 21 known epitopes from various malaria proteins in order to enhance immunogenicity. And these are known epitopes that for both CD4 and CD8. We put this multi-epitope string and the TRAP protein into viral vectors. So the two that we use are the simian adenovirus, ChAd63, and then we boost that response 8 weeks later with the modified Vaccinia ankara. And we've shown in a number of studies that this induces very potent T-cell immunogenicity. So the rationale for using chimpanzee adenoviral vectors rather than human adenoviral vectors is that it overcomes the problem of pre-existing immunity to human adenoviruses. These are genetically distinct from human adenoviruses. These are group E viruses. These viruses do not circulate in humans, and the seroprevalence is much lower, probably due to cross-reactivity rather than exposure in vivo. And these are some of the pre-vaccination titers that we observe. So these are children in the Gambia, 5 and under, adults in the Gambia, and UK adults. And you can see that whether you're looking in West Africa or the UK, the prevalence of neutralizing antibodies to the vector is very low. So we've been progressing over the last 6 years with this vaccine candidate through the various regulatory stages. We undertook a phase 1 safety study in UK adults, which demonstrated good safety and good T-cell and some modest antibody responses. And then we progressed to a phase 2A study in UK adults. And we have the great advantage within the malaria field that we can perform small-scale proof-of-concept challenge studies that involve vaccinating our volunteers with the regimen that I described and then exposing them to 5 infectious bites of falciparum-infected mosquitoes. We then follow those volunteers up for 3 weeks and look at the efficacy of the vaccine. And although it's only possible to do this in relatively small numbers for logistic reasons, it does mean that you can get a good proof-of-concept result before going to larger and much more expensive phase 2B efficacy studies in your malaria endemic target population. So, although I'm not a clinician, I'm just going to present a very small amount of safety data today. Basically, this is the adverse event profile for the adenovirus, very few systemic or local adverse events for adenovirus. MVA is always more reactogenic, but it's been given very safely to several 100,000 people now, and generally speaking, all adverse events resolve within about 7 days. So we see really good cellular and some modest humoral immunogenicity. So these are ELISPOT responses in recipients receiving adeno followed by MVA, and you see this great boost in the T-cell response as measured by ex vivo ELISPOT. The other thing that's great about boosting with MVA is you see a massive expansion in the breadth of the immune response. So typically after adenovirus, people might recognize 3 of the 6 peptide pools. Once you boost with MVA, that broadens to between 4 and 5 peptide pools generally, and we see responses along the breadth of the TRAP antigen. As I mentioned, the responses in this population, the IgG responses in this population are definitely detectable and increase above background, but they're not as strong as we see with some other malaria antigens. So within our sporozoite challenge study, we looked at efficacy, and you can see here in this Kaplan-Meier plot, the adeno-MVA group, we saw significant efficacy. 3 of our 14 volunteers were sterilely protected against malaria. 5 of the 14 showed a significant delay to parasitemia relative to the controls. And if you can produce a delay of 2 days in the time that it takes people to develop malaria symptoms, using modelling, we were able to work out that that actually equates to a 95% clearance or a 95% reduction in the number of parasites that emerge from the liver after 7 days. So that's a significant reduction in the liver emerging parasite burden. And here you can see the parasite, mean parasites per mil during the first 3 replication cycles after infection, after the emergence of the parasite from the liver at day 7. And over the 2nd and 3rd replication cycles, we see a significant reduction in the parasite burden in volunteers vaccinated with adeno-MVA, whereas in the adeno alone or the control group, we see no efficacy whatsoever. So in terms of immune correlates, we know that ex vivo ELISPOT doesn't correlate with protection. They're desperate for me to log into their network, aren't they? Sorry. We know it doesn't correlate with the ELISPOT response. There is some correlation between the liver-emerging parasite density and IgG antibodies to TRAP. But the strongest correlation is with CD8s expressing interferon gamma, and we've now published this. This is the first CD8 correlate of protection that's ever been published. And we see a very striking association between the level of circulating CD8s expressing interferon gamma and efficacy against malaria. And this is borne out from, and this confirms what's well known from both animal models and models in non-human primates, mouse models and models in non-human primates. So I put an extra slide into my talk last night because I've heard a couple of people say that one of the disadvantages of viral vectors is that you can't reuse them, you can't reboost with viral vectors. So we have shown and published that this is definitely not the case. It may be true for the human adenoviruses, but for the simian adenovirus serotype that we use, we've been able to reboost responses extremely effectively. So this graph shows T-cell responses after priming and boosting, and they gradually wane to sort of 600 by day 400. When you call people back somewhere between 6 months and 2 years after their initial immunisation, whether you reboost with adenovirus or MVA, you can boost that response back up to the original level that you saw after the first boost. You can never exceed the magnitude of the first boost, but it's certainly possible using the same viral vectors as in the original heterologous combination to achieve the same level of immunogenicity that was observed the first time. And we've used flow cytometry also to confirm that the phenotype of these reboosted T-cell responses is the same as what we saw after the first boost as well. We also look at the relationship between T-cell and antibody responses to the vector itself, so looking, for example, at the hexon protein, and responses to the antigen insert. And what we see is quite a strong positive correlation. So these are volunteers in that first challenge study that I showed you a moment ago. And we see anti-vector immunity as a measure of vaccine take. So what we generally see is that the greater the response to the hexon protein, to the adenoviral vector, the greater the response to the insert. So having completed a number of enabling trials in the UK, we then moved to our target populations in East and West Africa. So we have a partnership with the MRC Gambia unit at Sakuta, and also the Kenyan Medical Research Institute in Kilifi in Kenya. So I'm not going to go into much detail, but suffice to say this is the safety profile of adenovirus and MVA in Gambian and Kenyan adults. And as you can see, almost no adverse event profile for adenovirus at all, very few mild adverse events. MVA is always more reactogenic, but again, everything resolves very quickly within 7 days with a very acceptable safety profile. We also saw great immunogenicity in this cohort as well, similar to what we measured in the UK population. So having demonstrated safety and immunogenicity in East and West African adults, we then took our clinical trials back to the Gambia to do an age de-escalation and dose-finding set of studies. So these were 3 trials, children aged 2 to 6 years, 5 to 12 months, and 10-week-olds. And they received the same prime boost regimen with ME-TRAP that I described earlier. So adeno at week 0, 8 weeks later boosting with MVA, looking at safety and immunogenicity. And we had controls in each of these studies as well. So in the older children, we used rabies vaccine as a control. And just to control for the level of intercurrent illness in the younger age groups, we had unvaccinated controls for these. So this is the adverse event profile for 2 to 6-year-olds, 3 days after vaccination. Very mild adverse event profile, really well tolerated in these children. So looking at T-cell responses, so this figure compares ELISPOT responses in UK adults with Gambian adults, 2 to 6-year-olds in the Gambia, 5 to 12 months old, and then 10-week-olds. And what's immediately apparent is that once you get down to these older groups of children, the T-cell response seems to be much lower, and we were really quite surprised by this. particularly considering that it then seems to bounce up in the 10-week-olds, and we were really concerned that we might be seeing some hyper-responsiveness to the vaccine caused by malaria immunosuppression. And sorry. Okay. But if you've worked with pediatric blood samples, you'll know that young infants often have extremely high frequencies of lymphocytes when you do a differential blood count. And this just shows you the proportion of lymphocytes within whole blood in different age groups, in the same groups that I showed before. So as you get up to 5 to 12 months old and 10-week-old infants, the number of lymphocytes in the blood is much higher relative to the other white blood cell types. So what we did was intercalate both the lymphocyte count with the ELISPOT count per million PBMC to produce what we call a per mil readout, which, when you think about it from a physiological point of view, is much more biologically relevant, because it's ridiculous to compare the number of antigen-specific T cells in a baby on the same scale as it is to a human, when you know that the number of lymphocytes in a mil of blood is so different. And when you do this and take account of the lymphocytes, you then see that there's actually no difference between any of these groups, and the T cell responses across all the age groups were broadly comparable. We then looked at antibody titers, and here the results were quite different to what we saw with T cells. The responses in adults are admittedly low for IgG in the groups that we've looked at so far, and they're also low in the 2 to 6-year-olds. But when we looked at the two lower age groups, again, the responses were much better, and we saw really good induction of IgG responses in the youngest age groups that would be our target populations for immunisation with the malaria vaccine. Sorry. So the next stage of our vaccine development programme is to take this to an efficacy study in children. And this is being performed in a region of Burkina Faso. So this is the capital of Burkina Faso, Ouagadougou, and our efficacy study is down here in Banfora in southwestern Burkina, which is quite a beautiful part of Africa, actually, called the Cascades region. So we did a phase 1 safety study, an immunogenicity study, and 30 children there, and got approval to continue to a larger double-blinded study with 700 children enrolled in total, with a control group that received the rabies vaccine VeroRab. We immunised these children to coincide with the start of malaria transmission and followed them up with passive case detection. And we're now waiting for the first primary endpoint result data to be produced. And if you needed any convincing of why a malaria vaccine would be a useful tool in this part of the world, this is our malaria surveillance data from this region. So this is the number of malaria episodes in Banfora in the 6 months following on from vaccination with adeno and MVA. So this is a cohort of 700 children, and within those 700 children in 6 months, we had 1,100 episodes of malaria. So that's approximately 1.5 per child. And these are infants aged between 5 and 17 months, which is a frightening burden of malaria. So I hope that I've convinced you so far that adeno MVA ME-TRAP is safe and potently immunogenic for T cells. It induces a very broad response with multiple epitopes recognised in both European and African cohorts. And response to the simian adenovirus is much more potent than to human adenoviral vectors, and certainly overcomes the issues of pre-existing anti-vector immunity that have dogged programs with human adenovectors. We've shown a correlate of efficacy for this vaccine with CD8 T cells. And I didn't show it here, but we did a time to infection study in Kenya in 120 adults where we observed 67% efficacy, which for a semi-immune population is a fantastic result. We've seen great T cell responses and antibody responses in target age groups for immunisation in Gambia and Burkina Faso. And we have our efficacy data from the Burkinabe infants due very shortly. And of course, the other advantage of using adeno and MVA is the well-established GMP-compliant production methods, and so that you can adapt this adeno MVA platform to virtually any antigen you like, and we have programs for both flu, HIV, TB and malaria, as well as a number of other target diseases such as dengue running within the Jenner Institute at the moment. So, next steps for this programme, we're looking at a non-interference study in Gambian infants at the moment with EPI immunisations. The other thing that's really exciting is that we're now partnering with GSK. So the aim being to combine the excellent antibody-induced efficacy of RTSS with good CD8-induced efficacy from adeno MVA ME-TRAP, with hope of that combining those two approaches together, we can get the best of both worlds and produce something greater than the sum of their parts. And those trials are ongoing in Oxford at the moment. Hopefully, if that's successful, we'll take that to a study in adults in Burkina probably next year. So I've summarised about 6 years' work in 20 slides. So obviously there are many, many people involved in all of these trials, all of whom I'm grateful to, and particularly all of the volunteers that make these studies possible. So with that, I'd like to thank you for your attention, and I'll take any questions.
It's great that you're looking at combining the two different vaccination. Have you thought about actually making a multi-stage vaccine and using TRAP and maybe some other kind of liver stage or other
we've done
stage?
That. Yeah, so we did a study last year combining adeno MVA with CSP and ME-TRAP. And CSP in viral vectors does not induce any efficacy. It seems to be an antigen that works best by an antibody-inducing vaccine. And we've also done combinations of blood stage antigens. And as I mentioned before, viral vectors for blood stage doesn't really make much sense because viral vectors are very good at inducing T cells, and what you need for blood stage is lots of antibodies. So I think the best possibility for a viral vector multi-stage antigen is a liver stage component, which would be ME-TRAP, combined with a transmission blocking element. And the transmission blocking element of the malaria vaccine field is some way behind the pre-erythrocytic stage, but that work is in progress. Both ourselves and groups in the States and the Gates Foundation have been extremely positive in providing lots of money to push that forward. Yes?
Maybe I didn't get it right. I was just wondering, you mentioned that the ELISPOT did not correlate, but the CD8s correlated. So
Yeah.
the ELISPOT was looking at just purely CD4 soluble antigen response?
No, so ELISPOT looks at CD4 and CD8 and a number of other things. So the denominator for the ELISPOT assay is different to the ICS, and it's not until you pull out the CD8 specifically that you see that nice correlation, because you've got too many other cell populations kind of fudging the maths when you look at the ELISPOT response.
So I guess in your African infant trial then that you've just looked at ELISPOT right now, you would be looking again at the CD8s in those cohort samples, because you're not seeing big differences right now in yours.
Exactly.
ELISPOT.
So we do fresh immunology. I should say immunology on fresh PBMC in all of our clinical trials, whether they're in Africa or the UK. We do ELISPOTs and ICS on fresh, but that data is still in the process of being analysed at the moment. Thank you.